Dear student, this is an audio version of one of the fragments of a separation technique, precipitation of proteins and nucleic acid. Students, we know proteins and nucleic acids play an important role in modern biology. Thousands of proteins and different nucleic acids direct the whole cellular process. To study and analyze the number of proteins and nucleic acid at different stages of cellular development, it is necessary to extract them quantitatively. For this, precipitation is widely used. In fact, precipitation is widely used for the recovery of biomolecules. Precipitation is usually induced by addition of a salt or an organic solvent or by changing the pH to alter the nature of the solution. Now let us first see how proteins are precipitated. There are various methods of it, like it can be precipitated by salts, by using organic solvents, by using organic polymers, or by changing the pH of the solution, which we will discuss one by one. So to start with, we'll see first how the proteins can be precipitated by salts. The most common type of precipitation for protein is salt-induced precipitation. Salts such as ammonium sulfate and sodium sulfate are widely used to precipitate out proteins. Ammonium sulfate is the most widely used salt for the precipitation of proteins, as it is highly soluble, inexpensive, available in highest purity level, does not change the protein solution to extreme pH, and importantly, in most of the case, it does not denature protein. Ammonium sulfate can be used for the precipitation of total proteins at approx 90% saturation. Students, up to 20% saturation, ammonium sulfate precipitates particulate material and pre-aggregated and very high molecular weight proteins, and at 90% saturation, it precipitates almost all proteins. Now practically we'll see how protein gets precipitated by salts and what is the principle behind this. Proteins have polar amino acids such as glycine, serine, etc. Usually, in native proteins, hydrophilic amino acids are on the surface of proteins, whereas hydrophobic amino acids are buried. Attractive interactions between the nearby oppositely charged groups are ion pairs or salt bridges. Analysis has revealed that in folded proteins, 4 attractive ion pairs and 1 repulsive ion pair are present per 100 amino acids. Water, as a powerful solvent, interacts with this surface amino acids and keep them in solution. Along with this, protein solubility depends on several factors. It is observed that at low concentration of the salt, solubility of the proteins usually increases slightly. This is termed as salting in. But at high concentration of salt, the solubility of the proteins drops sharply. This is termed as salting out, and that means the protein precipitates out. During ammonium sulfate precipitation, The salt has to be added in a small amount under constant stirring to avoid accumulation of high concentration of salts. When large amount of salt is added to an aqueous solution of proteins, the salts require more amount of water for its dissolution. This leads to the competition for water molecules on the proteins. Completely ionized salts have more affinity for water molecules than protein. Hence, addition of salts takes up water molecules from the protein. Therefore, the ionic interactions between water molecules and proteins are reduced. And as a result of this, hydrophobic interactions dominate. The hydrophobic amino acid patches present in all the proteins attract each other and forms aggregates. These aggregates are nothing but the proteins in the form of precipitates. In salt precipitation, the ion appears to be more significant. But along with this, other factors like temperature, pH, and the most important, the protein concentration, affects the process to a larger extent. Higher ammonium sulfate is required for precipitating highly soluble proteins. There is another method of precipitating protein. Precipitation of proteins by organic solvents. Another method is the addition of solvent. Water-miscible organic solvents like ethanol, methanol, and acetone are good protein precipitant. The principle of solvent precipitation is different from salt. If there is a medium in which the dielectric constant decreases with the addition of organic solvent, the solubility also decreases, and hence we can expect precipitation of proteins. What is the methodology behind the precipitation of proteins using organic solvents? Students, we know water keeps protein molecules in solution by minimizing the electrostatic attractions between them. Water molecules reduces the strength of electrostatic attraction by a factor of 80. The principal effect of solvent is to reduce the dielectric constant of water during precipitation. Thus, solvent minimize the solving capacity of water molecules by reducing their dielectric constant. Hence, when the concentration of solvent increases, it brings about bulk displacement of water molecule. This allows strong hydrophobic interactions between proteins, and the hydrophobic patches of different protein molecules interact and aggregate. But when we use organic solvent as a precipitates, there are few important criteria which we should consider. First, solvent which we use should be completely miscible with water. Solvent should be available in highest purity. Another factor, it should not react with proteins. And very importantly, it should have a great precipitating effects. The most commonly used solvent is acetone and ethanol, but acetone is better protein precipitant than ethanol. Precipitation using organic solvent is done at 0 degrees centigrade or sub-zero temperatures in order to minimize the denaturation of proteins. In this method, solvent is cooled overnight and added slowly with constant stirring to ice-cold solution of proteins in cold condition. Then the precipitates are collected by centrifugation at 10,000 rpm for 20 minutes at 4 degrees centigrade. Advantage of using solvent precipitation is that it can be easily removed by evaporation. Another solvent which we can use for the precipitation is the organic polymers. Water-soluble, high molecular weight organic polymers are used to precipitate protein. PEG, that is polyethylene glycol, is widely used for the precipitation of proteins as well as for nucleic acids. Chemically, PEG are linear polymers of ethylene glycol. PEG is anionic and water-soluble, so it does not react with proteins and nucleic acids, hence produce minimal denaturation effect on macromolecules. Lower concentration of PEG precipitates high molecular weight proteins and vice versa. PEG with different molecular weight is available, but PEG with molecular weight more than 4,000 are very effective for precipitation. The most important factors for precipitation with this are pH and the salt concentration. Much lower concentration of PEG is enough when proteins are at their isoelectric points. Salt's concentration also influence the precipitation by PEG. The principle of precipitation of PEG is not well understood. It is assumed that PEG possibly brings about bulk displacement of water molecules like organic solvents and immobilize them. It may also involve in reducing the water activities like the organic solvent do. They have a stabilizing effect on biomolecules, which is possibly because of polyhydroxyl groups. The polymerized molecules favor the hydrophobic interactions among proteins and nucleic acids, which results in their precipitations. Another method is the isoelectric precipitation of protein. Solubility of protein is the result of polar interactions with water molecules and to some extent the repulsive electrostatic forces between the protein molecules themselves. If the repulsive electrostatic forces between the protein molecules are insufficient, then the protein molecules get together and forms the precipitate. This method of precipitation is done by changing the pH of the protein solution. This effect is due to the different functional groups on a protein. There will be some pH known as isoelectric point, where the net charge on the protein is 0. This is different for different proteins. When the pH of the medium is less than the isoelectric point of a protein, the protein molecules will carry a net positive charge. And when the pH of the medium is more, the isoelectric point of a protein, the protein molecules will carry a negative charge. As the like charges repel each other, the protein molecules stay in a solution at the pH above and below the IEP. But when the pH of the medium is equal to IEP, all the charges on the proteins are neutralized. Hence, in absence of any repulsive force, the protein molecules get together and precipitates out. So these are few commonly used methods for the precipitation of proteins. Next, we come to the methods of precipitations of nucleic acid. Nucleic acids are generally precipitated by ethanol, isopropanol, or PEG. The most commonly used solvent for the nucleic acid is ethanol. It is simple, rapid, and sensitive, as it can precipitate nanogram quantity of DNA and RNA in few minutes. Residual ethanol can be easily removed by air drying or vacuum drying the pellets for few minutes. DNA concentration as low as nanogram can also be easily precipitated under standard conditions. For effective precipitation of nucleic acids, monovalent cations like ammonium acetate or lithium chloride or sodium chloride or sodium acetate is added before the addition of cold ethanol. Buffers containing high concentration of EDTA should be avoided during ethanol precipitation, as it gets precipitates along with the nucleic acid. RNA is also efficiently precipitated with the above mentioned cations with 2.5 to 3 volumes of cold ethanol. Like proteins, there are different chemicals which can be used for the precipitation of nucleic acids. Let us see how nucleic acids can be precipitated using isopropanol. In this, large volumes of DNA samples can be efficiently precipitated using 1 volumes of isopropanol at 0 degrees centigrade and incubating the solution for approx 30 minutes along with the monovalent cations, and the remaining isopropanol can be later on removed by evaporation. Another chemicals is PEG. PEG, a water-soluble organic polymer, is used for the precipitation of DNA, and we have seen it is even used for the precipitation of proteins. High molecular weight DNA can be precipitated with low concentration of polymer and vice versa. PEG enhances hydrophobic interactions among DNA molecules and bring them closer in the form of precipitates. Another, precipitations using polyethylene amine, shortly called as PEI. PEI is a highly branched synthetic cationic polymer, very effective in nucleic acid precipitation, especially from cell extracts. PEI at 0.3% precipitates 90% RNA and 96% DNA at pH 7. But be careful when using PEI, as it is carcinogenic. Precipitations of protein can also be done by using another chemical called as CTAB, that is cetyl trimethyl ammonium bromide. CTAB is a cationic detergent, especially used for the extraction of DNA from DNA fragments. CTAB binds DNA strongly and forms an insoluble complex. This chemical has a strong affinity for DNA as it carries positive charge, and DNA carries negative charge and hence precipitates out DNA. In this method of precipitation, salt concentration is very important. When the concentration of salt is above 1 molar, no precipitate is formed. But when it is less than 0.2 molar, all the nucleic acid is precipitated. And if the concentration of salt is maintained between 0.3 to 0.4, then small DNA fragments and oligoprimers are not precipitated. Hence, CTAB provides a suitable way of extracting different size of DNA. Another important property is it also solubilizes membrane, and therefore it is used in the extraction buffer for the lysis of cell. The last chemical which we can use for the precipitation of protein is TCA, that is trichloroacetic acid. Trichloroacetic acid precipitates nucleic acids and protein efficiently at a concentration of 5% to 10% at 0 degrees centigrade. Generally, 20% to 50% stock solution of TCA is mixed with equal volume of the sample, which finally gives you the concentration of 5% and 10%, and then incubate at 0 degrees centigrade for 30 minutes to give complete precipitation. So dear students, I wish you have learned different methods of protein and nucleic acid extraction in a simple way. Thank you.